anaerobes. So for anaerobes, you have to be very thorough. It has to be without oxygen. So you need to provide them special conditions where, or a special jar where there is no oxygen. When you culture anaerobes, you use sodium hydroalcoholate in the media so that there is no oxygen. All the oxygen is absorbed, and the whole jar is devoid of any oxygen. We also take palladium as a catalyst, where we discuss about it also absorbs oxygen. They also use sodium borohydrate and sodium bicarbonate. So there are different agents. The main aim of all these agents is to have a jar which is devoid of any oxygen. Then we also talked about the way you culture or microorganisms that need low oxygen. You put them in the standard jar method. And then we talked about that what is the enrichment culture, what is the selective media, what is the differential media, and how a media can be selective and differential, or it can be both. We also talked about the enrichment culture technique, that how do you enrich for a particular microorganism even when they are present in lesser numbers. Okay, so now we are going to move ahead towards more practical aspects of microbiology, where we are going to talk about how to define a pure culture. Okay, so you will be doing a lot of these things, but unknowingly. Okay, all of you must have done cloning and other things, but when you do cloning, you pick one colony and do mini prep. Have you done that?
Yes.
In your second semester practicals, I think you have not done that. Is it? Am I right?
We didn't do, yes, sir.
You did that. Did it?
No, we did not. Did not.
You did not. Okay, yeah, I asked you before also. So you did not do that. But for example, if you see a plate with lot of colonies, you take one colony from it and you inoculate, isn't it? That part you have seen?
Yes.
Okay, but so what do you mean by one colony? Okay, how do you define a colony? How do you define a pure culture? It's very important microbiology term. Okay, always remember that when I take a culture from a complex source like a soil or water or urine sample or your blood sample, you will always have a mixed culture. What do you mean by mixed culture? It means there will be a lot of microorganisms present there. And now from this lot of microorganisms, how do you get a pure culture? What do you mean by pure culture? Pure culture is the one, it means I have a microorganism which is actually coming from a single bacteria or a single microorganism. Okay. So we will try to define this microbiologically. Okay, so how do you obtain a pure culture? Okay, so what you do is you cultivate your bacteria on a solid medium. Okay, plates like this, all of you must have seen them. Okay, so you normally do that on a agar plate. Okay, what is agar actually? It's an algae extract with polysaccharide nature, and very few bacteria can degrade it. Most of the time, bacteria will not degrade it, and your plate will be 1.5 to 1.8% agar. Okay, and what is a colony? This is a very important definition. Okay, you should not forget this definition. This colony is a population of a bacterial cell which is coming from a single cell. It means the way I plated it, this colony, although there will be multiple bacteria, but has come from a single bacteria. Okay. So, then what is pure culture? Pure culture, it contains only one species or strain. Okay, so if you will not have, if it is an E. coli colony, it should be only E. coli colony. If it is an E. coli OH157 colony, it should be E. coli OH157 colony. If it is an E. coli OH158, it should be 158 colony. Okay, it means I am having only one species or strain in that culture. Okay, and what is a colony? It's a population of cells arising from a single cell or a spore or from a group attached. Okay, and one colony arises from a one colony forming moon. Okay, so specimens like pus, sputum, foot will have different microorganisms, and the common way to isolate is from doing a streak plate method. Have you done streak plate method by any time? By any chance?
Yes.
Okay. So do you know how does the street plate method work? Do you know the principle? Or do you know just how to do it?
It's a step with sequential dilution. Right? Once you streak
Yes.
it, and then the sample gets diluted as you streak it further.
Yeah, you're right. So what you do is, basically to explain to all of you is, you have a you have a mixed culture. Okay. So you make a smear like this. Okay. Now what you have done is, you have now actually you have started adding lines to it. As you are adding lines, you are actually diluting your culture. I had a mixed culture here, but when I add a line, I'm actually diluting it. Okay. From now, 1,000 bacteria present in this culture, I may have, I would have diluted 10 times. It becomes 100 or so. When I put another line, I will again dilute it 10 times more approximately. Okay. So I'm diluting my culture. And then again, when I speak a line, I dilute it. Okay. So it means in a plate, I'm sequentially diluting the culture. Okay. And you can simply ask why you need to do streak plate. Just dilute the culture and plate it. No, we can do like that also. Okay. But sometimes what happens is, you can actually see, you can get multiple colonies also. Okay. So each colony may be coming from a different bacteria. When you dilute your culture, suppose you can say that you can dilute your culture in the LB itself and then plate it. Why you need to streak it? Okay. It may be possible that you will not get any colony, because I do not know the dilution factor. Okay. Or it may be possible that even if I dilute it based on thinking, okay, I have diluted the culture 10 times, but still it may be very concentrated. I may not be able to, do it properly. I will still get a lawn on the plate. Okay, but when you are able to sequentially dilute it, you will be able to see the gradation in the number of colonies. If my culture is too dilute, I may get, the first spot itself, I will get lot of colonies. But it will be a single, I will get individual colonies. Okay, so when I do not know the dilution and I still want to have a pure culture, I just start diluting it on the plate itself. Okay, this is a very lazy method to do it. I can make dilutions also, which I will explain to you, and put it on a plate. But I need 4 or 5 plates. But with the streak plate method, I can do it in one plate itself. I can take a concentrated culture, smear it, and then make lines. There it will be enough diluted. I will get individual colonies. It may happen in the first one or in the last one. Okay, so that's the advantage of having a streak plate. It's the most common method for bacterial isolation. It is the object is to reduce the number of cells being spread. It's a solid surface. So what you're doing is you are actually diluting it. You can do anything you want. There is no specific pattern. Some people like to do it like this. Some can just do it like this, or some can do it like this. Okay. Basically, your principle is to just dilute the culture from the original culture on a plate itself. Okay, so you can have any number of beautiful pictures you want to have on the plate, and you will get an isolated colony. Okay, so we have actually learned that, okay, we can grow culture, but the important part is how to preserve the culture. Okay, you can grow, but what will happen? Bacteria will continue to grow. The important part for you is to know how to preserve your culture. Okay. Now it depends what is your need. For example, you are doing a project in the lab. Now what has happened is all of you are taking classes. Okay. And now in the morning, in last night you inoculated your culture. And in the morning what you did was, now you have a class at 9:30. You cannot go for plasmid isolation. Okay. Now what will happen? Now you have to preserve the culture. But you have to preserve the culture so that you can do it in the afternoon, isn't it? You have to preserve it so that the bacteria doesn't overgrow and lies. You have to preserve for 4 to 5 hours. Okay, so that's your one need. Okay, you have done the experiment, you have inoculated it, but you have class in the morning, you cannot do a mini prep in the morning. You cannot do a plasmid isolation in the morning, you can only do it after lunch. So how to preserve for those 5 hours? Your next experiment could be, okay, you inoculated the culture and now you have midterm exams coming up. You will not be able to do it for 2 weeks. Because for 2 weeks you had midterm exam. Now the problem is you have to preserve your culture for 2 weeks. I need a longer preservation time. Okay, now your third need could be that okay, you did mini prep, you found okay, this is my positive stone, and you want to preserve it for indefinitely, so that if you come back after a year or so, you are still able to grow the bacteria. Okay, so it means you want to preserve for hours, you want to preserve for days, and you want to preserve for years. So you have 3 requirements for preservation of bacterial culture. Okay, and based on this, your method of preservation of bacteria will be different. Okay. Okay, so if you want to just preserve it for hours, you put it in refrigerator. You put it 4 degrees. For example, you inoculated your bacteria yesterday. You have a class, you want to do it in the afternoon. You just put the bacteria in freeze 4 degrees centigrade, and you will find there will be no problem. Okay. For example, now you want to go to deep freeze. Okay. When you want to preserve for years. Okay. So how do you do it? You take your bacteria, put it in glycerol, and you freeze at -80. Okay. Now what you do is, so this is known as deep freezing. You store your cultures at -80 degrees with the help of glycerol. Now this another method which can actually last for decades. Where you freeze your culture, you dehydrate your culture in vacuum, and now you get a powder of bacteria, and that you can last for decades. Okay. You must be seeing a method of lyophilization. Now, doctor gives you gut bacteria. Doctors will normally prescribe you gut bacteria as tablets. All of us take that now, isn't it? You think your stomach is not well, you take antibiotics. The doctor also gives you some capsule which is probiotic. Have you heard about probiotic capsules? What are probiotic capsules? Probiotic capsules are actually lyophilized bacteria. What the company does, the company grows the bacteria, freeze it at -80, does a vacuum drying, and now they get a powder of bacteria. Now once you eat that bacteria gets the proper nutrients, that's the proper temperature, it starts multiplying in your blood. Okay, so probiotics capsule where you have lyophilized powder actually is a bacterial lyophilized powder which has live bacteria. But what has happened is they have kept the bacteria at -80, frozen the bacteria at -80, vacuum dried it so there is no water now, and you get a powder which can last for decades. Okay, so this is known as probiotic. Normally people are using it as probiotic, and it's the best method to preserve bacteria. You can do it even after 10-20 years. You take the bacteria, put it in the media, it will start growing. Okay. So that's the best method. The lyophilization method is almost the best method. But if you want to use it for a week, okay, you don't want to lyophilize it. You don't want to undergo that process. You can just take the bacteria, keep it 4 degrees, and do your work. Okay. So again, remember, you do not follow the best method always. You follow what do you need. Do you want to preserve the culture for short periods of time or for long periods of time? Your experiment should be very clear. Okay. Do not go for complex experiments. Go for simple experiments, and you have to go for the need. Okay. If you can always say, no, ma'am, I can just lyophilize the bacteria and do it after a week. What's the problem? But is it really worth your time? Is it really worth your effort? because you have to freeze the bacteria, do a vacuum, right? That process itself will take 1 day for you. While keeping in the fridge will take 30 seconds. So you have to weigh your options, and that weighing of your option is your rational thinking, and you should be very clear about the basics. Okay. So these are the methods of preserving bacteria. You can refrigerate it for short periods of time, you can deep freeze it at -80 and culture it for years, or you can lyophilize it and it can last decades. Okay, so how does, so again when you take this bacteria and put it in a media, the bacteria starts to, multiplying. Okay, the process of bacterial division, all of you have learned, so I'm not going to go into the detail of that. But basically what happens is you have a bacteria, your cell wall and membrane will start to grow inward, and then it will be actually, there will be a cross wall formed and you will get two bacteria. Okay. Now what is important in terms of bacterial division is that from one bacteria you get two bacteria. Okay. So this is very helpful when you want to find out the generation time. Okay. So it means that each time bacteria divides, the population doubles. If I am going from one bacteria, then based on the generation time, I will have two bacteria. Now based on the generation time, I will have four bacteria. Okay, and now the type varies from one bacteria to another bacteria. What is it? It means by the time the cell elongates and comes into two, this is different from one bacteria to another bacteria. Okay, so what is generation time? The time taken for doubling to occur is called the generation time. In what time your one cell becomes two cell is known as your generation time. Okay, and under optimum conditions, the doubling time is very typical for each bacteria. Okay, and remember this is greatly affected by your culture conditions. Okay, for example, Salmonella typhoeria can grow in the lab in 20 minutes, but in the body it may take 20 hours. Okay, let me explain this to you a bit more.
Ma'am,
Yes.
just a doubt about the previous topic about storing the bacteria. Is it
Yeah.
okay if we switch the storage method midway depending upon our needs, or will it not give the same efficiency?
See, if you keep your bacteria 4 degrees for a week, the bacteria will start dying, isn't it?
Yes,
So
ma'am.
you will not have that efficiency.
Okay.
So if you take a bacteria, there will be some bacteria, but it will take long time to grow. Okay. So 4 degrees will be only good if you want to store that for only 4 to 5 hours. I cannot keep the bacterial culture for a week and expect, okay, everything will be fine. Bacteria will not be happy. Okay, remember about what I talked about when I talked about the psychotrophs, mesotrophs and thermophiles.
Yes, ma'am.
Because what is happening at 4 degrees? If it's a bacteria like a E. coli or a bacteria, it will grow very nicely at 37 degrees. But at 4 degrees, it will still be alive, but it will not be the optimum condition. And when there are no optimum conditions, it becomes a problem for the bacteria. It will tend to die, it may not adapt, it becomes stressed. Okay. But within 4 to 5 hours, nothing will happen and then you can continue. 4 to 5 hours is fine. But if you want to keep it for a week, it's impossible to keep it for a week.
Then is it fine if we switch the after using storing it for 4 to 5 hours? Is it fine if we switch the method and then freeze dry it for like a week?
That should be okay. People do not actually prefer it sometimes, but like when you want to do a normal experiment, like for example, now I can give you, for example, you have made the clone now and you want to freeze the bacteria after 4 to 5 hours because of your class. It works perfectly fine. Okay. It will also depend sometimes on bacteria to bacteria, but mostly it should be okay. Like if you go for 4 to 5 hours, keep it 4 degrees. It's not the best, but I can say still you will have 95% survival.
Ma'am,
Okay.
ma'am,
Yeah.
Why the glycerol dip is required in deep freezing?
Okay, that's a nice thing. Why this all is required? Okay. Now, what will happen if I do not put? You normally will freeze your culture in. You will grow your LB. Okay. So now you have grown your culture in LB. Then you freeze it. What will happen now? It will become ice. Your cytoplasm will also become ice, isn't it? So if I put a glycerol, it acts as a cryoprotectant basically. So it doesn't allow the formation of ice actually. You do not get ice crystals which will lyse the bacteria. So you normally add 10 to 15 glycerol so that it acts as a cryoprotectant and there is no formation of ice crystals. So your cytoplasm is fine, and then the bacteria doesn't lyse or it doesn't lyse when you put it again in the media, because there is no formation of ice crystals. But if you do not add glycerol, there is formation of ice crystals, and now once cytoplasm will also become ice now. And what will happen when again put the bacteria in the medium? Your cells will lyse because you will have the formation of ice. So glycerol basically acts as a cryoprotectant.
Yes, thank you.
Yeah. Anything else? Okay. So what I was talking about generation time. Okay. Generation time is, for example, if you very well you will very well know about E. coli. Okay. E. coli, if I put it in LB media, I will get from one cell to two cell, I will get in 20 minutes. But if I put in a chemically defined media, it may become 1 hour. So based on your culture conditions, you can have a different generation time. If I provide more nutrients, it means I will have a better generation time for bacteria. Okay. Similarly, for example, if I have a pathogen, okay, for example, if I have Salmonella typhimurium, if I grow in a bacteria in a plate where I give everything, then the bacteria will grow in mostly 6 hours. Like in 6 hours, one bacteria will become two bacteria, and then 2 becomes 4, and then 4 becomes 8. But when I grow in mice or in humans, it will take 20 hours to grow. Okay, that's why when you have an infection in your body, you tend to have a lag period. Okay, and then the bacteria multiplies very fast. Okay, so your generation time is constant for a bacteria. If I put E. coli in LB, it means it will be always 20 minutes. So in a particular condition for a particular bacteria, E. coli will be having 20 minutes, and the same culture, Salmonella typhimurium, may have 6 hours. So again, this is very typical of bacteria. E. coli or Salmonella typhimurium it doesn't mean it will be 6 hours and then it can become 20 minutes. That is not possible. Generation time is very typical, but you can manipulate your generation time based on your culture condition. Whether you provide a chemically defined medium, you provide a rich medium, you can have differences there. Okay, so these are very two important facts to remember about generation time. It's very important for you guys. You may think this is very similar, but if you have you prepared competent cells? Have you seen how preparation of competent cells? You normally wait for the OD of 0.4, isn't it? So normally, how do you calculate when you get a point? So what is I will explain you what is OD actually. I have started talking before. So I know that if I put one bacteria now, it will become 2 bacteria in 20 minutes. So I can calculate my number of bacteria. Okay. So it's very important for calculations. Okay. It's very important when you have an infection. For example, if I have an infection, you must have seen. Like you must have seen your blood culture report, urine culture report. They write CSU per ml. How do they get to that number? It's also based on the generation time. Okay, so these are very important microbiological parameters which will define the type of bacteria and the infection you have. Okay. Okay, so what you have is your generation time. And generation time is defined as the time required to divide for population to double. Okay, average for bacteria is 1 to 3 hours. E. coli generation time is 20 minutes. So 20 generation in 7 hours, one cell becomes 1 million cell. Okay, in 20 generation in 7 hours, you will have 1 million cells basically from one bacterial cell. Because the generation time is 20 minutes. But if I have another bacteria which is like Salmonella, and if it has a generation time of 6 hours, your final number will be very different. The generation time is very important to calculate the final load of a bacteria in a population. Okay. If I want to plot it as a log phase, graph, then I will have an initial lag phase and then there is an exponential phase. Okay. And if I put it on a semi-log scale, it will always be straight line because from one bacteria is becoming two bacteria, two will become four. So you will always have a straight line. Okay. But on an arithmetic scale, you will have this will be a lag phase because I will not be able, my numbers will not be in the exponential. This figure is very important. So this number is n is equal to 2 over n is very important. Based on how much cells I have inoculated here, I can calculate what will be my cells of this after 10 hours or 5 hours or 24 hours. Okay, so this equation is very important for microbiology that n is equal to the power n. Okay. Now, what are the different phases of growth of a bacteria? Okay, most probably all of you have learned this, but just to understand in terms of generation time, what you have is lag phase. Lag phase doesn't mean that the bacteria is not growing. I plot it over time and log scale. Okay, so in this case, I will not see any growth of bacteria. Okay, my graph is very flat here. But remember that doesn't mean that my bacteria is not growing here. My bacteria is growing here, but my numbers are very constant. Okay, and I will explain to you in a bit why it is there. Then what you have is an exponential phase where I will see a constant increase in the number. Okay, this is the time, this is the most active phase of the bacterial replication, and generation time is actually really in the log scale. Okay, this is the exponential growth phase. What you see is a stationary phase where again my bacterial number has not increased. My bacterial number it remains the same. And then what you have is a death phase where I can see the decline in the number of bacteria. Okay. So in this case, what is happening actually? Okay. So if I have to think about it, here I have extra amount of nutrients in the media because I have just inoculated my culture. I may have inoculated 1 or 2 bacterial cells, and what I am seeing is there is no increase in number, but my nutrients is very rich, my media is very rich. Okay, here the exponential phase is the best phase because I have the balance of number of bacteria also and balance of amount of nutrients. Nutrients are perfectly balanced, bacterial number are perfectly balanced, and what you see is the exponential growth. Now what has happened in this case is, now there's lot of bacteria that has happened, my nutrient conditions have started depleting. Okay, so what is happening is, even my bacteria is multiplying from 1 to 2, but because of the bacterial death, because my nutrients have become very less, because of the starvation conditions, I am also seeing the death of the bacteria. Okay, so when you see the death of the bacteria, then my numbers are almost equal. Bacteria is multiplying and some bacteria is dying. So I don't see the change in the numbers of the bacteria. Although there will be high amount of bacteria, but still my numbers will not change. And now in the last phase, what has happened is my nutrients have become really less. So most of the time your bacteria is dying, and what you see is even your replication has stopped happening. Okay. So this is the death phase of the bacteria. Okay. Now, just to make you understand what is your lag phase. Okay. So in the lag phase, what you see is basically you see when you add one bacteria, why you do not get a reading here? Why my graph is very constant here? Because you should not take it if my bacteria is not multiplying here. It's only because I'm not able to see, judge the growth of the bacteria. Okay, if do a microbiology experiment, you will understand that, like when you inoculate a bacteria for 2 hours, there will be no turbidity, isn't it? You don't see any turbidity for 1 to 2 hours. Doesn't mean my bacteria is not growing. It means I'm not able to see. The numbers are very less here. So I'm not able to judge the count of the bacteria. Okay. So that is not the lab phase. The replication is happening, but I'm not able to see the bacterial growth because the numbers are very less. And once the numbers have achieved a critical amount, then you see exponential growth, and then you see a turbidity very high. Okay. So if you want to make a competent cell, you normally take an OD of 0.4. I will explain what do you mean by OD actually. And that's the time when you see within 20 minutes, you will have 0.4 to 0.8. The numbers will from 0.5 million, it becomes 1 million in 20 minutes. That's the exponential phase happens. Okay. Because nutrients are enough, bacterial numbers are enough, and I can judge the bacterial growth. And in stationary phase, my numbers do not change, my turbidity will be very high, but I will not be seeing anything because the nutrients have started becoming less. Okay, and then you have the death phase. Okay, so for any bacterial growth, what you see is the S phase first. Okay.
Ma'am,
Huh?
is it right to put it in these words that lag phase we see mostly a straight line because relative to the other stages, like the exponential growth stage, we see less growth. So like less number of bacteria. So relatively,
Yeah, because you will not. If you read it, I will explain. I will come to that point. If you read it, your OD or your method will be still 0. Your count doesn't change. Okay. And so it's not. I cannot say. Do not mistake that the replication or the bacterial division is not happening. Do not mistake with that. Normally, we try to mistake that there is no bacterial division is happening. It's just the numbers are too low for us to detect by eye. Okay, it may be possible that I take, for example, I can give you an example. How will I judge it like this? Okay, you should not believe me. How will I prove it? That the bacterial division has taken place, but I am not able to judge it. Okay, so for example, now I have taken a flask which is just empty, and I have inoculated the bacteria, and I start monitoring it. Okay, so I see there is no increase in the growth of the bacteria. You normally see by turbidity, isn't it? You will see, okay, I can see bacterial growth is turbid, media is turbid. Okay, but how do I prove it in the lab phase the bacteria is growing? What I can do is I can take 1 ml of the culture and plate it on a plate. After 20 minutes, I can take another ml of the culture and again I pellet down everything. I pellet down 1 ml of the culture. And then you will see that you start seeing the increase in the number of colonies. But when it sees over the whole LB media, I may not be able to see it because the numbers are very less. Okay. Did I answer your question? I think I got deviated.
no, it got answered. Thank you.
Yeah. So normally like division is taking place, but your numbers will be very less. So you will always see a clear media. Because I'm coming to that point only. See, every technique where you want to measure the growth of the bacteria has its limitation. Okay, so I was coming to that point that how do you measure? Like the way we had this doubt that lab phase, what is happening? How do you measure the growth of the bacteria? Okay, so there could be very chemically defined parameters to do that, or it could be a physical method to do that. Okay, so my next point is how do you measure it? Okay, you know there's a lag phase, log phase, stationary phase, but how do you measure the growth of the bacteria? Okay, there could be a direct method. It could be a plate count method, filtration method, MPL method, or microscopic count method, or there could be indirect method, which is turbidity method, metabolic active method, or dry wet method. Okay. Again, remember each method has its own advantages and disadvantages. Okay, we have to the method which you prefer should be your choice because based on your experiment. Okay, The most frequent method is your plate count method. Okay. So basically the most frequently used method is very simple. You take your bacterial culture, take and do a spread plating method. Okay, and what you see in the next on the next day, basically what you will see is your single bacterial colonies. Okay, so what are the assumptions I am taking here is each colony originates from a single bacterial cell. Okay, another one is my original inoculum is homogeneous and there are no cell aggregates which are present. Now what will happen is I will measure only viable cells here. I will not, because this bacteria which is dead, I will not be able to, they will not be able to grow in the plate, so I will not be able to measure them. Disadvantages is it normally takes 24 hours for the colonies to appear, and if I have less number of colonies, less than 25, it's not good. If I have more colonies, it's not good. So you should perform serial dilution. Okay. Before going to that, I will just explain you without the help of slide is the spread. This is the most common method which you use for counting the your bacterial group. If you get good number of colonies, you will do that. Moment you set up your transformation, if you see good number of colonies, you will be very happy. Okay, so normally plate count is the best method. Okay, but you assume there that each colony is coming from a single bacteria. It's a pure culture. Okay, that's the assumption there. The disadvantage, advantage will be it will only count viable cells. Okay, it could be only possible that the bacteria is not able to use that media. Okay, so this is the disadvantage. If I ask you for a bacterial population in water, okay, some bacteria may not be able to grow, they will be dead bacteria. I will not be some, but if I ask you, okay, what is how much viable bacteria are there in your culture? This is a good method to do that. But if I just want to get the total number of bacteria, irrespective whether viable or non-viable, then it's not a good method. Okay, because plate count will give you only the cells which are alive and are able to grow on that particular medium. Okay, so that's but it's the most common method and it's the most easiest method. Now what will happen is, for example, I took a plate and now I have lot of colonies. No, I get lot of dots. Very difficult to count. Okay, and if you have, for example, only 2 bacterial colonies, you do not know the contamination coming from your culture, you do not know that also. Okay, so how do you work around those conditions? If you have too many colonies, the best way is to do a serial dilution. Okay, I have 1 ml culture. I got 20. Suppose I got a lot of the colonies. So what I will do is I will take 1 ml from this can, dilute to 9 ml of water. So, for example, my 2000 colonies now becomes 200 colonies. Again, I diluted 10 times more, it will become 25 colonies. So, based on my dilution, I can do a serial dilution and then do a plating, and then I can get respectable number of colonies, and then I can count CFU per ml, colony forming units per ml. And this is known as the serial dilution method. So what you have is like when you have taken the original inoculum, what you see is you plate 1 ml and then what you see is a lawn of colony. But then you take 1 ml and put in 9 ml of media and then you plate 1 ml. Then you started seeing a decrease in colony. Then again you dilute it. You still get some colony, but again there is a lawn. And then again you dilute 1 ml. You see a respectable number of colonies. So this is a plate you should be able to count. Do not believe these 3 plates for your counting. Always believe these plates for counting. The number should be between 25 to 250 in a plate for a good count. Okay. Even do not take this plate to be a good count. Because there are only 4 colonies. So a number between 25 to 250 is a better plate to calculate your CFU per ml. Okay. So how do I do it? So in this plate what you have is 32 colonies, which is come with the dilution of 1:1000. Okay. So in your original one, you will have 32 into 10,000 colonies, which is 320,000 bacteria per ml. Okay. Because I am plating 1 ml here, And after a dilution of 10,000, I am getting 32 colonies. So if I concentrate my culture 10,000 times, I should have in 1 ml 320,000 of bacteria. Are you able to understand this calculation? It's a simple calculation, but all your pathology reports depend on this calculation. When you write, okay, your blood culture is negative for bacteria, they calculate this number, CHU per ml. They do serial dilutions and calculate it. So CHU per ml is really important. How much of blood? If you have a CHU per ml which is like 250 colonies, doctors will really prescribe you antibiotics. If it is 1-2 CHU per ml, it will not, they will not, it will say it will go away. Okay, so all your pathology reports with bacterial infection deals with this type of CHU per ml. So this is a very important number. So did you understand how to calculate it? It's a number of colonies multiplied by the dilution, and if you have plated 1 ml, it becomes per ml. Okay.
Now? Now I have a doubt.
Yeah.
Why do we need, in experiments, why do we need a specific OD? Why can't we just test the OD of our prepared culture and then serial dilute it to get how much ever dilution we want?
Okay, that's a very nice question to understand. See, you do not want to work with you always want to work with a culture which is in the exponential phase. You do not want to work with a culture which is in the stationary phase because remember the bacteria are dying here. So that's why if you even calculate, see, now what will happen when you take a culture from a stationary phase, when you plate it on a media, you will not be able to get the exact number. Because probably your 10 to 20% bacteria, dead bacteria. So your total number of bacteria will not be reflective on what you have got in the plating. You will always get less numbers because in the stationary phase, you will have a population of a bacteria which is dying. Okay, so normally when you want to set up an experiment, like when you want to do a protein production or when you want to do a competent cell preparation, you do not want to work with the lab phase because the numbers are very less, and you do not want to work with the stationary phase because you will see the dead bacteria, which is not good. So you always want to work with the exponential phase, which starts with the OD of 0.4. I will talk about what is OD basically, but that's the time when your culture hits the exponential phase, and it's the best phase because you do not see any death of the bacteria. Bacteria are really happy, multiplying in 20 minutes, and what will happen is, and your numbers are also good for making your work. You don't want to work with less number. You can work in lab phase also, but the problem is your numbers will be very less. So how do you work with less bacteria? You also want to have good amount of bacteria to do your work, isn't it? You don't want to work with 10, 20, you want to work with at least 50,000 of bacteria. And by the time you keep it, you will have enough number of bacteria for your experiment. So that's why when you want to do any experiment, you want to do it in the exponential phase, not in the lab in the stationary phase. What you can do is, you can take a culture from a stationary phase, do a serial dilution, and then allow it to grow for 3 hours. Then the bacteria which is alive in the will be in the exponential phase.
Okay, thank you.
Okay, and I'm going besides the point here, but there is a way you can maintain the culture in the log phase. Do you know that method? Have you heard about it? For example, just think about it. If I ask you a simple problem that I have to really keep the culture in the log phase, what should I do then? It's a very simple thing. Just think about it.
Maybe
Supply
freeze it.
nutrients. Keep on supplying.
No, I have to have a continuous culture which I want to keep it in the log phase. I'm growing it. I don't want to freeze it. Freeze it is like I'm storing the culture, but I want to have a regular culture. When kept in the log phase, I don't want a dead phase. Yeah, one of you was answering that.
Like keep adding the nutrients to the media again and again so that the nutrients will
Yeah, so that's the way your actually fermentation principle works. What you need is you need to have a constant nutrient. Okay, but what you also do is also remove some media because you cannot increase your media content. Okay. If I have 1 liter of media, I cannot make it if I keep on adding continuously. It will become 1 liter, 2 liter, 10 liter. Okay, so how this happens in the industry? Because industry doesn't want to deal with the stationary phase culture. Especially when you make antibiotics or anything, like if you go to a this is a part of basically industrial microbiology, where the industry wants everything should be in your exponential phase. They don't want to work with lab, they don't want to work with stationary phase. So what they do is, and remember there is oxygen deprivation condition. So what they do is they have a bigger flask, they allow the bacteria to come to log phase, and once this is the maximum log phase, they will remove 500 ml of the media and add 500 ml of the fresh media. Okay, so now again my nutrients will become, it will not be less, and now again I will maintain the log phase. For example, instead of 3 hours, I can maintain for 20-30 hours. Okay, so that's the way industry normally tend to maintain the bacteria in the log phase. You replace and provide fresh media. You replace and provide fresh media. Any more questions, sir?
Ma'am, in that case, won't the number of bacteria keep increasing? Do they also compensate for that in any way to keep the number also constant?
The number is very difficult to keep constant actually. Numbers they don't keep constant because they want to have more number of bacteria.
Okay, yes.
Okay, the purpose is exponential phase you get. So the purpose of your from industrial microbiology basically they want to increase the number of bacteria. So that's the way they can do it. But the problem is if you have a 10 liter, they cannot make it to 10 liters or something. So they generally tend to replace some, and add media, something like that. So that's the fermenter, fermentation industry that works. Okay, so that's a serial dilution. Okay, and how can you do that? That's a pour plate method. People do not use it normally, but that was an Asian method which was developed. So what you do is, I will explain to you, and then the other one is just spread plate method. Okay, what is basically in a pour plate method? We do not follow this, but you should know this. So what you take is you have a plate, and then you add your melted agar, and then what you see is you also add your culture to it. Fine. So you in an empty plate you add your culture, then you add a melted agar, this solid, and what you get is colonies. Okay, this is the pour plate method. What is happening in case of spread plate method is you have plate which is already poured with your solid medium, which all of you are doing. For example, LB agar. You take a small amount of culture, plate it, and then you spread plate it, and what you get is an individual colony. Okay. Can you think what is the disadvantage of this method? Disadvantage of a pour plate method?
Colonies might form in a bulk at some places. Wouldn't be any colonies there.
Possibly that is one of the problem. This could be another problem, which actually I always face when I pour a LB agar plate. You do not know how hot it is, isn't it? Sometimes you think, oh, it's very hot. Sometimes you think, oh, it's become cold, it should not solidify. You always struggle with this decision, or you don't struggle, you are better than me. Isn't it? I always struggle. Is it too hot or is it less hot? It is cold because see, when the agar starts solidifying, it becomes a problem. It just solidifies very fast. So what will happen if I add a very hot LB to the bacterial culture? Okay, the bacteria may die. So to have the perfect balance is sometimes difficult. Okay, and if I have, for example, 10 plates, I'm adding in the one plate, my temperature was fine. By the time I reach the 10th plate, my media solidified. Now again I'm in a problem. And I will not be able to swallow properly. Okay, so if your swallowing is not proper, then what you will get is the aggregate of colonies. Another problem is if your media is too hot, then it will kill the bacteria. And third problem is I will get colonies in between the agar plate. If you do it on spread plate method, you will get colonies on top, and then you can pick up a colony and inoculate it. But when you put a pour plate method, you will get colony immersed in your agar, which is sometimes not good. Okay, so this is the disadvantages of a pour plate method. That's why people do not follow pour plate method. So always, when you want to do a transformation, you have to make LB agar. So again, it's a very practically not a viable method. Where you can store your nutrient agar plates for a long amount of time, you can keep it for months. You can plan the experiment 1 month before and then do it. But with the LB agar plate, you go, my autoclave is not working, you cannot do the experiment. So it's not even a practically viable method to do that. So normally people do not follow pour plate method. So disadvantages is not pour plate method, not useful for heat sensitive organisms, and colonies appear under agar's surface. Okay. Spread plate method is the most preferred method. Its colonies will be on the surface and will not be exposed to the melted agar. Okay. Okay. Let me just finish with this. For example, how do you do that by filtration method? Okay. Let me just finish. Okay. Why do you use filtration? Okay. For example, I asked you yesterday for an endosphere technique. Okay. You told me I have a plastic degrading bacteria. So if I take a 1 ml of water, my number of samples will be, if I ask you, okay, what is the total count of bacteria? You may not be able to get any colony from there. If I just take 1 ml. Okay. But to get respectable amount of bacteria from a water sample, what you need is 1 liter of water. Okay. Now how do you plate 1 liter of water? It's very difficult. You cannot, use 1 liter because you can only plate, maximum 1 ml. Even people do not plate 1 ml. They plate only 100 to 200 microliters. And if I ask you to centrifuge 1 liter of water, you will not be able to see anything. It's very difficult. So it means how I have to get the total count in 1 liter of water, very dilute sample. How do I concentrate it? Even if I pellet it down, I can centrifuge the bacteria. I may not be still able to see a good bacterial pellet. So the method of choice will be a filtration method. Why? I will use a membrane which is 0.22 micrometers. And most of the time your bacteria is greater than your 0.22 micrometers. So what I can do is, I can now filter 1 liter of water on a 0.22 micromembrane. So all the bacteria will come onto that membrane. Okay, so now I have a membrane which is rich of bacteria. I have concentrated all the bacterial population on a membrane. Now I can do two things from there. I can take that membrane and just invert it on a bacterial plate and I should be able to get colonies. That's one way to do it. Or I can just, add some medium to the bacterial membrane, shake it well so that all the bacteria which is stuck onto the membrane comes out and I plate everything. So from 1 liter, I can resuscitate them in 1 ml of media and then I should be able to get the good growth of the bacteria. Okay. So with filtration, what I have done basically, I have concentrated my dilute sample to a respectable amount so that I can plate it on a plate. Okay, so that's the way people use the filtration method to get a proper amount of colonies forming unit per. Okay. So when you have dilute samples, filtration method is a method of choice to find out the number of bacteria in a population or to measure the counting of bacteria. So, like for example, if you have a large sample, then you normally filter it on a 0.22 micrometer membrane, and then you transfer it on a petri dish and incubate and count the colonies. Okay, so that's the way you do a filtration method counting. Other things I will take in the next class. Are you clear about this? Any doubt?